So I promised to talk about the other two types of autophagy in a minute and then kind of forgot, so I'll start with that. So we were talking about macro autophagy as a means by which potentially large portions of the cytoplasm are enclosed into a double-walled structure that fuses with the lysosome to destroy everything, including the original membrane from the autophagosome, as a result of the proteolytic or hydrolytic activities of the lysosome. There is a completely distinct mechanism, so distinct that probably it should have a different name, but it doesn't. It's referred to as chaperone-mediated autophagy, and that's really more of a sorting rather than trafficking event in which a protein in the cytoplasm is brought to the surface of the lysosome and then translocated in. Very reminiscent of mitochondrial sorting, for example, and like that, it features prominently a cytoplasmic HSP70 type chaperone. The C is for cytoplasm, HSC70, and a trafficking or sorting motif that leads to this, the attachment of this protein to the surface. So this is the cytoplasmic surface, this is the lumen of the lysosome. That leads to the recruitment of the subunits of yet another translocon, in this case the lysosomal one, and the translocation of that protein here into the lumen. And once again, there's the Brownian-Rashett mechanism, this time with the luminal HSP70 type, and then specific involvement of an HSP90 type in the lumen of the ER. So that would lead to the translocation into the lumen of a protein from the cytoplasm, and its fate might be to function in the lysosome, or it might be to be destroyed. But that would be chaperone-mediated autophagy. So before going on to the third type, I'm not going to talk about this one at all. So the third type I'm going to talk about is microautophagy. But I wanted to mention another thing that goes wrong with aging when we get older, and that is that all of these processes become less efficient. So in the case of macro, and really both chaperone-mediated and microautophagy, one of the causes of inefficiency is the pH in the lumen of the lysosome. It starts out when you're young and healthy, being around 5.5, and it gradually gets higher and higher. It gets more closer to neutral as we age. So all those degradative processes tend to work less well the older we are. And as far as chaperone-mediated autophagy is concerned, these components tend to become less abundant with age, so that we get less able to bring in proteins by this kind of sorting mechanism. Okay, so moving on then to the last type of autophagy I want to talk about, the so-called microautophagy, is a reaction between vesicles and the lysosome. It's very poorly understood, but I did want to, well, first of all, to say that, I see that question mark, and this is 2020, so this is not something that is known even today. But notice what's going on here is it's not exactly fusion, and it's not budding the way we're used to, where something buds out into the cytoplasm from a vesicle. This is budding into the lumen of something, producing a region of the cytoplasm that's now enclosed in a membrane, and all of that is in the lumen. So in this case, microautophagy could bring in relatively large amounts of material from the cytoplasm, not obviously as big as this with macroautophagy, but bigger than a single protein. So whatever is in here now is going to be subjected to those hydrolytic processes and low pH and degradative activities that we associate with the destructive capacity of the lysosome. Okay, so this question mark, there is some evidence to suggest what it might be like, and that's because there's a very well understood phenomenon that is shown here that is not the same as this. This is the endosomal lumen, which is not the same as the lysosome, although it may become the same thing before too long. But what's shown here is the specific details of that inward budding process, and the fact that it involves specific green things on this diagram. These are integral membrane proteins that are going to mediate this internal budding process to produce these so-called intraluminal vesicles. I'll talk more about these in two panels. So we're going to talk about MVBs, multivesicular bodies, and essentially this is what is being formed in this panel specifically. But for now, I want to move on to some other aspects of endocytosis related to signaling. Now, to do that, I want to stick with what we've been talking about, and that is endocytosis related essentially to nutrition. That's what this process is. It just has an additional wrinkle. So the nutrient to be gobbled up in this process is cholesterol. Cholesterol is enclosed in a protein complex to make it soluble in the extracellular space. That complex is the low density lipoprotein. It is recognized by integral membrane protein on the surface of, let's say, a fat cell, the so-called LDL receptor, and internalized using the stuff that we've been talking about. So that would get us all the way through to the early endosome. Now, there's nothing wrong with this receptor at this stage, so sending it to the lysosome would be wasteful. So instead, it's recycled back, and this is a very rapid recycling, to the plasma membrane where it can bind more LDLs and keep the process going. Meanwhile, cholesterol goes through the entire lysosomal pathway, gets degraded and released for subsequent use. So this is essentially pure nutrition in the sense that there's no regulation involved here. As soon as the LDL receptor appears here, it's shipped right back, and it's continuously present. So as long as there's LDL, there will be LDL receptor to take it up, and that's the way that system works, and there's no need to turn it off at any point. But by contrast, we can think of a slight variation on this theme involving receptors that undergo signal transduction reactions that need to be turned on, but also obviously need to be turned off too. So we can imagine that as a diagram, say down here, where here's the plasma membrane and here's the receptor in question. Like so. And we can just start with the system in the off state because there is no ligand for it. So let's just imagine that this is, say, a growth factor receptor, just so that we can maintain it in a predictable form. And if we do that, then there will be the arrival of the growth factor out here in the extracellular space, and the receptor will bind to it. And as a result of the way growth factor receptors do, there will be an oligomerization of those receptors. And the only importance of that is that it sort of leads into the next phenomenon, which is the formation of a coated pit, clathrin-coated pit. That clustering will help in the overall formation of that pit. So it's kind of a convenient additional benefit to that mechanism that's involved in signaling in the first place. So that would look something like this. Now, while that's going on, this growth factor receptor is doing what it's supposed to do. That is signaling to the cell that it's time to divide, to proliferate. So that signal is going to be conveyed to the cytoplasm, and I like to think of these things as acting like beacons that broadcast into the cytoplasm. So this process of budding doesn't change that at all. The cytoplasmic region is still cytoplasmic and can still signal just fine, and that's true even with the internalized endocytic vesicle itself. So we have an orientation in it that might look something like this, in which the receptors can still be clustered. There's nothing to prevent that, and they can still be signaling. There's nothing to prevent that either. So, and this can go all the way through to the endosome, and the orientation, if you fuse this cargo vesicle with the early endosome, which is basically the only thing that we've talked about so far anyway, then the cytoplasmic region of that receptor will still be sticking out into the cytoplasm, so potentially you can get that beacon still working. So in that sense, this endosome or this trafficking vesicle can be referred to as they are in the business as signaling structures. So we refer to signaling endosomes as those that specifically possess an active receptor for signal transduction. And I'm not going to talk more about that until we've talked about receptors and signal transduction specifically, but I did want you to be aware of the connection between endocytosis and signaling reactions, and we'll talk more about that later. There's another signaling component to this process, and that would involve subsequent fates of that receptor. So if we follow this process through in this direction and stipulate that the receptor in this case is not recycled, it is brought into the lysosome or the endolysosome along with the growth factor, then you can see that this overall process will terminate the signal because everything has been chopped to pieces. So this is a classic way by which cells regulate signal transduction cascades: off, on, and then off again as a result of the removal of receptors from the surface. So we can think of that, the product of that series of reactions as this, that is a plasma membrane that doesn't have receptors on it. So this cell becomes desensitized temporarily due to the absence of those receptors, and the only thing that's going to make it respond to that growth factor again is making more receptors. So that happens over time, and eventually you'll get the overall transcription, translation of and sorting and trafficking of new receptors to make the cell respond again to those signals. But in the meantime, it can be thought of as being refractory to growth factor presence, and it doesn't respond to those. And that's a key component in the regulation of a whole lot of signal transduction pathways. That mechanism goes by a name. It's referred to in the business as receptor-mediated endocytosis. So I won't say anything more about it now, but when we talk about specific receptor pathways, in particular the G protein coupled receptor pathway, we can talk about ways in which that process can be turned on basically actively, directly with components of that regulatory process.